viewed by endoscopy. In this technique, a flexible microscope tube is inserted into the mouth or anus with fiber optics providing illumination. Here, the endoscope has been inserted into the lower intestinal tract via the anus. It's difficult to examine the surface of the gut in any great detail because peristalsis is constantly changing the shape of the gut. But its highly convoluted appearance can be seen. Endoscopy only shows us gross intestinal structure. It tells us little about how the tract functions. In this program, therefore, we'll be using a range of techniques which allow us to study digestion, and in particular, the functional anatomy of the digestive tract. Dr. Nick Reed, Sheffield University.
Now, when you've finished your meal, I'm going to be coming along and taking a few breath samples, and this will tell us how quickly the meal has passed through the gastrointestinal tract. Now, digestion is the process whereby the complex molecules of protein, fat, and carbohydrate which are present in this meal are converted to smaller molecules which can then be absorbed. And absorption is the process whereby these smaller molecules pass from the inside of the intestine across the intestinal wall into the blood, from where they pass either to stores or go directly to the cells for the purposes of energy. Now I'm going to have my meal. But I'm not going to eat it. This represents my stomach. The meal is transferred into the stomach. Mashed potatoes and some baked beans and sausages. And I always like to finish up my meal with a nice cup of tea. And finally, when the meal enters the stomach, gastric juice is added. And gastric juice contains hydrochloric acid and also an enzyme called pepsin. We put the lid on here. Now the whole mixture is churned up by the stomach so that it is of a homogeneous consistency. So the food is mixed up with gastric juice in the stomach, and it then passes from the stomach into the duodenum, which is the first part of the small intestine. And this is represented by this beaker here. Now, in the duodenum, the process of digestion takes place. The food mixture is combined together with bile, which enters the duodenum from the gallbladder, and pancreatic juice containing enzymes is also added to the food. The food is now digested into small molecules which can be absorbed. Now, to give you some idea of the capacity of absorption which takes place in the small intestine, this large volume of food and digestive juice mixture is reduced during a 5-hour passage down the small intestine to this much smaller volume of material here. Now, clearly this model gives us some impression of how the upper gastrointestinal tract works, but it gives us no impression at all of the anatomy of this region. And for this we have to use other techniques, and the technique which is most commonly used in man is the radiological technique of a barium meal. Okay, now that's the stomach there. The big portion filled with barium is the fundus of the stomach. Try to just follow it on down through the stomach.
Would you like to lie on your back?
There we are. Now we can see the body of the stomach. You can actually see some air going down through the body of the stomach. And this is heading towards the antral region. So the barium is still up in the fundus of the stomach. The body is below the fundus of the stomach.
Keep going to the right side.
And if we look carefully, we might be able to see a contraction.
A little more to that.
Well, we can't see this at the moment because it's tilted around the other way. There, the barium is running down into the body of the stomach here,
Right over.
towards the antrum. It's filling a bit of the antrum around there at the moment.
Over and get down.
But turning the patient round, so we're getting it different, getting all of this at different angles at the moment. Okay. Now we've turned the patient right over on his back, and the stomach is now looking in the other direction. The barium is in the body of the stomach, and if we look across a little bit, we can see some of the barium's gone through into the duodenum and is just going round the duodenal loop. Now that area right on the right-hand side is the C-shaped duodenal loop, which is inverted here because the patient's over lying on his stomach. Now, if we look carefully, you can see a contraction, an indentation in the antrum, which is heading towards the pylorus. A little kink. Now, it's gone at the moment. We might see another contraction start in a minute. There it goes. You see the kink? The kink is heading towards the pylorus. There it goes. A kink, an indentation pushing towards the pylorus. There it goes. It moves quite slowly, and it's heading towards the pylorus, and it pushes a little bit of barium out into the duodenum. There it goes, around the duodenum there. That's really quite nice. So you can see a gastric contraction. It only occurs in the antrum, this gastric contraction. It only occurs in the distal portion. Here comes another one, and it's sweeping down. Let's just watch this one. It sweeps down towards the pylorus. It's pushing the barium ahead of it. You see, into the duodenal cap, that area at the top, and then round the duodenum a little bit. Now, this is over on his back, so the stomach's facing the other direction. We're going to go up and have a look at the fundus now, first of all. That's the fundic area. That's the reservoir portion of the stomach, right at the back. Okay. Now, you can see all this barium coming round at the bottom here is, in fact, going around loops of duodenum which go behind the stomach. So there's the fundus of the stomach at the back. There's the body, which is mainly outlined with air, with just barium outlining the outside of the stomach. There's the antrum at the end, and there's a contraction going down the antrum again. You can see it the other way. This is the duodenal loop, the C-shaped duodenal loop. That's the area that pancreatic juice comes in the middle of this C. And here's barium going backwards and forwards along the jejunum, just beyond. There it goes. That's a loop of jejunum. It goes along there. I'm going to point to it a little bit here, if I could. Duodenal cap, pylorus, duodenal loop, right the way around here, nicely distended. The pancreatic juice comes in this area here, into the duodenal loop. Now, he's lying on his stomach, so this is all back to front again. But oh, that's nice. You can see things moving there. Look at that. That's beautiful. That really is nice. See it really whizzing around there now. That's lovely. That's nice.
What's that?
Well, that's going down the small intestine. It's going down towards the ileum, in fact, that. Oh, look at that whizzing around there. That's beautiful, isn't it? You know, who says gastroenterology isn't beautiful? That's really nice. It's poetry in motion, that, you see. Lovely. So that's going down towards the ileum. Oh, this is a lovely shot, really. You couldn't wish for anything better, that. The peristalsis is pushing it right the way down the intestine. Here it goes. And you can see the gas, the air in the bowel, outlining other loops down there. So that's nice. This is coming down towards the ileum here. So the ileum almost sort of keeps the food there for some time. It whizzes quite quickly through the jejunum, but it seems to gather in the ileum.
Radiology tells us about the gross functioning of the tract in living animals. To understand its coordination, we need to turn to experimental animals. Professor Tim Scratchard, Sheffield University.
Digestion begins in the stomach, but the major part of it occurs in the duodenum and in the small intestine. Just look at the anatomy of the upper part of the gastrointestinal tract. Here we have the stomach, which is divided into two functional parts, the body and the antrum. The antrum leads through to the pylorus and into the small intestine, which I will lift up here. This is the first part of the small intestine, the duodenum. And you can see in the duodenal loop the head of the pancreas. This discharges its secretion into the duodenum through a duct, which is about here. It is so small that it can't be seen by the naked eye. The bile duct empties with it, which is a very much larger structure. If we now lift the stomach up and look at the structures underneath it, we can see that the pancreas extends across the abdomen, under the stomach, to as far as the spleen. Replacing the stomach, we can see again our body and antrum. On eating a meal, food enters the body, which acts as a receptacle, and through a nervous reflex, this relaxes to accommodate the increased volume. This relaxation is called receptive relaxation. The meal distends the body to stomach, and an additional reflex sets the antrum in motion as a pump. If we turn now to the pig stomach, we can see this adaptive, the adaptive nature of the muscle coats. This is the muscle coat of the body of the stomach, and you can see it is fairly thin. This is the muscle coat of the antrum, and you can see that it is fairly thick and in line with its function as a pump. So the antrum pumps food out into the duodenum. In life, or in the decerebrate animal, we can see these movements beginning at the junction of the body of the stomach with the antrum when the nerves of the stomach are stimulated. Deep waves of peristalsis pass down across the antrum, through the pylorus, and into the small intestine. And as each wave passes down, it carries with it a small bolus of food. Now, what controls stomach emptying? The vagus, on the one hand, induces stomach motility. On the other, we have inhibitory factors acting from the duodenum, both nervous and hormonal. This is the duodenal brake, and its function is to prevent the overloading of the duodenum in terms of volume and acid. The vagus also stimulates the production of acid, and when acid gets into the small intestine, it liberates the hormone secretin. If secretin is injected into the arterial supply of the isolated pancreas, you can see that it produces a large volume of isotonic sodium bicarbonate. And this happens also in the intact animal. When the vagus liberates acid, it also has terminals on the acinar cells of the pancreas which liberate enzymes. And when the secretin stimulates the flow of pancreatic juice, these enzymes are washed out into the duodenum. Enzyme stimulation is continued by the hormone cholecystokinin pancreazymin, CCK-PZ for short. And this is liberated by protein digestion products from the wall of the duodenum. Thus, the coordination of pancreatic secretion in the digestive sequence is brought about largely by those factors which control the emptying of the stomach.
Once in the duodenum, the major part of the digestive process occurs. Proteolytic enzymes from the pancreas continue the protein digestion that was initiated in the stomach, proteins being broken down into peptides and amino acids. Carbohydrases, also from the pancreas, break down carbohydrates into simpler sugars. Fats are emulsified by bile, produced in the liver. This lowers their surface tension and makes it easier for pancreatic lipases to break them down. But in order for the body to utilize the products of digestion, they must be absorbed from the lumen of the tract.
Now, absorption is the process by which the small molecules of digestive food pass across the intestinal lining into the bloodstream. During the 5-hour passage of food down the small intestine, this volume here of food plus digestive juice is reduced to this small amount of food residue here. Now, how is this prodigious absorptive capacity possible? The small intestine is covered by myriads of finger-like processes called villi, looking rather like the tentacles of a sea anemone. And if we look closely at the surface of one of these villi, we can see that it is packed with small intestinal cells or enterocytes. Similarly, the surface of each enterocyte is covered by finger-like processes called microvilli. And the villi, together with the microvilli, increase the surface area of the small intestine by some 3,000 times. Now, there is one condition called celiac disease in which the villi are completely absent from the small intestine, leaving a completely flat surface. Patients with this condition are unable to absorb food properly from the small intestine. They lose weight, they develop diarrhea, and they also have various mineral and vitamin deficiencies. Equally important in terms of how quickly food is absorbed in the small intestine is the length of time that it is in contact with the intestinal lining or epithelium. If the food is in contact with the epithelium for an insufficient length of time, it will not be absorbed properly and diarrhea may well result. Now, we can measure the length of time that food is in contact with the small intestine by means of the gamma camera.
The gamma camera measures gamma radiation emitted from food to which a gamma source has been added. This source has a short half-life and has no deleterious effect on the patient.
Now, the patient has eaten the meal together with the radioisotope some 2 hours previously, and we can now study the distribution of the meal containing the radioisotope by means of the gamma camera. This shows the stomach here. This area here is the small intestine, and this blob here is the large intestine. Now, you can see that some 2 hours after eating the meal, some of the meal is still present in the stomach, some of it is present in the small intestine, but the majority has already gone through into the large intestine or colon. Now, it is possible to store this image on videotape and study it on a television screen. Now, the stomach here empties into the small intestine here. This is the upper part of the small intestine called the jejunum. This is the lower part called the ileum. This in turn empties into the colon seen here. Now, it is possible to count the amount of radioactivity over the area of the small intestine, and this can give us a figure for the length of time that the food containing the radioisotope remains in contact with the absorptive surface in the intestine. This ranges from about 2 hours to 5 hours in normal subjects.
There's relatively little cellulose in the diet of humans, and it's just as well because we aren't able to digest it. But many animals are herbivores, and this poses a problem, since they too lack the enzymes necessary for cellulose digestion. Ruminant animals solve this problem by having symbiotic microorganisms present within their stomach. Consequently, the design of the stomach is very different from in humans. I've got a model of the sheep's stomach here, and it consists of 4 separate chambers. The esophagus enters the stomach between the region known as the reticulum and the rumen. And in the reticulum and the rumen, we have symbiotic microorganisms, and these ferment the cellulose material, and the end product of the fermentation process is fatty acids, and these are absorbed across the wall of the rumen. Digested food passes from the rumen and reticulum into this bulbous region, which is called the omasum, and the omasum strains the food and prevents any large undigested material passing into the abomasum, which is the true stomach, analogous to the stomach in humans. And here, gastric juices are secreted, and the gastric juices contain hydrochloric acid and pepsin, which is an enzyme that's responsible for protein digestion. And any undigested material can be digested in the abomasum, and also the symbiotic microorganisms are digested, and these provide an important source of nitrogen for the sheep. Food passes from the abomasum into the duodenum, which is the first part of the small intestine, and the rest of the intestine in sheep is similar to that in humans. Now we're going to look at a real sheep's stomach. I've cut open the sheep's stomach, and here we can see these structures, which are the papillae on the surface of the rumen. And these increase the surface area of the rumen, which is quite important because of its absorptive function. This honeycomb structure is the reticulum. It's known as honeycomb tripe, and tripe eaters may well recognize this structure. The reticulum has a similar function to the rumen, but the reticulum also serves as a receptacle for debris, nails and stones which sheep inadvertently eat. The third chamber of the sheep's stomach is the omasum, and the wall of the omasum has a number of leaf-like folds, and these folds are believed to help to keep back large food particles and prevent them from entering the abomasum. And the abomasum is this pinkish structure, and you can see these plicated walls on the surface of the abomasum. And the abomasum leads into the duodenum. The esophageal groove is this region next to the esophagus, and this has a very important function in young ruminants, because in these animals they don't feed on grass, they're receiving milk from their mothers, and milk bypasses the rumen and reticulum because the lips of the esophageal groove come together to form a tube. So milk passes directly via the esophageal groove into the abomasum. But cellulose digestion can be a difficult process, and there are often large particles of grass present in the rumen which can't be digested by microorganisms. And these large particles are periodically regurgitated, that is, they are passed back up from the rumen through the esophagus to the mouth, and they are remasticated. That's called chewing the cud. And this remasticated food, along with large quantities of saliva, are re-swallowed and pass back down the esophagus to the rumen. And the saliva, which is alkaline, performs an important function because the fatty acids which are produced by the microorganisms lower the pH inside the rumen, and the saliva helps to neutralize the acidity, and this is quite important because otherwise the microorganisms would be killed.
He's lying on his stomach, so this is all back to front again. But oh, that's nice. You can see things moving there. Look at that. That's beautiful. That really is nice. You see it really whizzing around there now. That's lovely. That's nice.
What's that?
Well, that's going down the small intestine. It's going down towards the ileum, in fact, that.